السلام عليكم ورحمة الله وبركاته. بسم الله الرحمن الرحيم. Dear students, today we are going to take the fourth lecture in cytology. We're going to talk about one of the non-membranous organelles, which is the cytoskeleton. What do I mean by cytoskeleton? It is a complex network of microtubules, micro and intermediate filaments, together with some proteins to link them to each other and to the cell membrane, forming a framework called microtubular lattice. Here is an EM picture showing the microtubules and microfilaments. Here is a diagrammatic picture showing that there are two types here of tubulin, alpha and beta tubulin joined together. Alpha and beta tubulin joined together form protofilament. Here is the alpha and beta tubulin joined to another one and as well to the other one, forming what's called protofilament. 13 protofilament joined together, forming one microtubule. This is in a TS section or cross section of one microtubule formed of 13 protofilament. One microtubule formed of 13 protofilament. Again, alpha and beta tubulin join together, forming one protofilament. 13 protofilament form one microtubule. If you count this TS section, you'll find that's formed of 13 protofilament. By definition, microtubules are fine tubes of constant diameter 24 nanometer but unfixed length. By LM difficult to be seen except by using immunofluorescent technique. What do I mean by immunofluorescent technique? If we consider the microtubule is an antigen, we are going to label antibody with fluorescent and put it with the microtubule. For demonstrating if there is microtubule, the antibody will join the antigen with the fluorescent. I'm going to see the microtubule. By EM, they are fine tubules formed of alpha and beta tubulin, as we saw in the previous diagram. Alpha and beta tubulin join together and each form the protofilaments. 13 protofilaments form the microtubule. The length varies according to the polymerization of the tubulin molecule. The diameter is constant, but the length depends upon the polymerization and depolymerization of the tubule. This is directed by Microtubular Organizing Center, or MTOC, which contains another type of tubulin known as gamma tubulin. So, dear students, what do you know about the tubulin? We have three types: alpha, beta, and gamma. The alpha and beta join together, forming protofilament. 13 protofilament will form the microtubule. What can control the polymerization? The presence of gamma tubulin in the microtubular organizing center. Here is the nucleus. Here is the microtubular organizing center, which is near the nucleus. You can see here rings. These rings called the gamma tubulin. From these gamma tubulin, alpha and beta tubulin join together and polymerize, forming protofilament. 13 protofilament will form a microtubule. Forms of microtubules, we have either dynamic with continuous assembly and disassembly, causing, for example, reshaping of the cell. What do I mean assembly and disassembly? Adding or moving the tubulin molecules. As we said, the diameter is constant, but the length depends upon the polymerization and depolymerization. Stable. No, they are stable. No polymerization or depolymerization occurs, as in the wall of the centriole, cilia, and the flagella. What is the function of the microtubule? Determination of the shape of the cell, intracellular transport of the macromolecules. Inside the cell, the molecules move. As we remember, the secretory vesicle which arise from the mature phase of the Golgi apparatus move toward the surface of the cell in order to open on the cell membrane by exocytosis, for example. Formation of centrioles, cilia, and flagella. Formation of mitotic spindle during cell division. Cancer chemotherapy used to arrest the cell proliferation in tumor by interfering with this microtubular assembly. These are this is one pair of centriole and this is another pair, and this cell is in dividing condition. This is the metaphase. Mitotic spindle arises from the centriole in order to divide the chromosomes. If this is a somatic cell and these are 46 chromosomes, 46 chromatine moved here and 46 chromatids moved there by the action of the mitotic spindle. If the cancer chemotherapy arrests this mitotic spindle, so this cell will not divide. What is the structure formed by stable microtubules? The centrioles. The centriole is a non-membranous organelle and this is responsible for cell division. It appears a cylindrical structure composed of highly organized microtubules. By light microscope can appear by special stain known as iron hematoxin. Dear students, do you remember when I said this stain before? When did I mention this stain before iron hematoxin? Yes, in the mitochondria. So it can be demonstrated with iron hematoxylin and appears as two dark bodies near the nucleus. By the electron microscope, two cylindrical structures perpendicular to each other and surrounded with a matrix of tubulin known as the centrosome or the microtubular organizing center. Here is one pair of centriole. This is one centriole and this is another centriole. So this one pair of centriole formed, they are cylindrical in structure, formed of well-formed microtubules. Here are three microtubules together forming triplets. If we count these triplets, we're going to find that they are 9 triplets. The wall of the cylinder is formed of 9 bundles of microtubule, and each bundle is formed of 3 microtubules known as triplets. If we multiply 9 by 3, so one centriole is formed of 27 microtubules. The microtubules in the triplet, they are arranged as A, B, and C. TS shows that it's formed of 13 protofilaments, while B and C, they share protofilaments together, so B and C are formed of 10 protofilaments. This is a longitudinal section of the 9 triplets, and this is TS section of the 9 triplets. As you see here, it's formed of each triplet A, B, and C. A is formed of 13 protofilaments, while B and C sharing together formed only of 10 protofilaments. Again, this is 9 triplets. Each triplet is formed of A, B, and C. A is formed of 13 protofilaments, while B and C is formed of 10 protofilaments. What is the function of the centriole? During S phase of interphase, the centrosome duplicates itself. During mitosis, it moves to pools of the cell and become organizing center for microtubules of the mitotic spindle. And I'm going to show you how. Share in the formation of cilia and flagella. Dear student, this is the cell cycle. The cell, in order to divide, it should enter the cell cycle. G1, the cell grow. S stage, DNA duplication, as well as centriole duplication, and G2 I'm preparing for mitosis. The mitosis occurs. In the S phase of the interphase, duplication of centriole occurs. Why? In order to form the mitotic spindle. This is an EM picture showing one pair of centriole lying perpendicular, another pair of centriole perpendicular. These are the chromosomes, and these are the mitotic spindle in order to divide the chromosomes. Here is the basal body of the centriole. Is formed of 9 triplets. Okay, this is the TS section, and this is the LS section of the centriole. Microtubules containing organelles. We first discussed the centriole. Secondly, the cilia. What is the cilia? It is a motile hair-like process projecting from the surface of the cell. How it is formed? Centriole duplicate 1,000 times and migrate to the surface, and this is during embryological development. The cell that should be ciliated, during its development, centriole duplicate 1,000 times and migrate to the surface. This is the cell surface. The basal body of the cilia is equal in structure as the centriole. With the same structure of the centriole, 9 triplets. This is the basal body of the cilia. 2 microtubules from each triplet. 2 microtubules from each triplet, so they are called doublet from each triplet. These 2 microtubules, A and B, grow outside, forming finger-like process, pushing the cell membrane in front of it, forming what's called the axoneme. Dear students, we have 9 triplets. If I have 9 triplets and 2 doublets from each triplet move, so I'm having 2 by 9, so I'm having 18 microtubules projecting in the surface. In addition, in the center of the shaft, 2 microtubules appears known as the singlets. From where it appears, as we said, there is a microtubular organizing center. 2 centrioles will polymerize in the center of the axoneme, forming what's called the singlets. The third microtubule, which is the C microtubule, okay, move downwards, forming the rootlet in order to fix the cilia in the cytoplasm. This is LM picture of the cilia projecting at the surface of the cell. By LM, the cilia appears as hair-like striation on the surface of the cell. By EM, the finger-like cytoplasmic projection from the surface of the cell covered by cell membrane containing 9 peripheral doublets in addition to 2 singlets. So 9 by 2, 18, and having 2 singlets, 18 plus 2 equal 20 microtubules. The basal body had the same structure of the centriole, 9 triplets, 27 microtubules. The rootlets is downward growth of the outer microtubule C in the basal body, 9 microtubules to fix the cilium to the cytoplasm. This is a TS section in what? Guess. Is this, we said that the cilia is formed of 3 parts, basal body, shaft, and rootlet. What this section? Is it basal body or shaft or rootlet? It is shaft. Why it is not basal body? Because we said that the basal body is formed of 9 triplets. These are 9 doublets. Okay, and I'm having 2 singlets. So this is the shaft or the axoneme of the cilia. Okay, 1, 2, 3, 4, 5, 6, 7, 8, 9. These are the 9 doublets. This is A and this is B microtubule. Okay, these are the singlets. If I take this doublets, this is A and this is B, A joined to the Next, B, by what's called nixon. By what's called nixon. A joined to the singlets by radial spoke. A joined to the singlets by radial spoke. And A contain two dynein arms which contain ATP. Microtubule A of each doublet connected to microtubule B by nixon. Next, microtubule B by the nixon. The central microtubules or the singlets by the radial spoke, connected to dynein arms which motor protein using energy from ATP. Again, this is shaft, basal body, or the rootlet. It is the shaft. In addition, they are doublets, 9 doublets, and this is the singlets. There is the cell membrane. The basal body is not surrounded with cell membrane. Again, this is A containing 13 protofilament, B containing 10 protofilament, A connected to the next doublet B by the nixon, A connected to the singlet by the radial spoke, A connected to dynein arms. What is the function of the cilia? The cilia move in a wave-like manner to move particle or fluid of a tissue surface in one direction as the respiratory system and the female genital system. In the respiratory system, we have in the trachea, bronchus, there is cilia and small bronchioles. There are in the trachea and in the bronchus, in the upper respiratory passage, there are cilia. These cilia move particles in one direction in order to avoid entry of any particles to the alveoli. In the female genital system, as in the fallopian tube, moving the ova from the ovary to the uterus in order to be fertilized. It can be modified to receive stimuli as rods and cones in the retina. The cilia, the flagella is a long cilia forming the tail of the sperm, and the tail of the sperm is making the sperm able to move toward the female genital system. This is the flagella, which is a tall cilium. Okay, have the same structure as the shaft of the cilium, but longer and larger. The flagella form the tail of the sperm, direct and facilitate the movement of the sperms. A condition known as immotile cilia syndrome. The cilia is not moving, and this condition causes male infertility in addition to chronic respiratory tract infection. These are the cilia which present in the normal respiratory passage. If the cilia present and do not move, this will make the respiratory tract continuously infected, and in the male, the flagella will not be able to move, so there will be male infertility.